Welcome friends. In this video I'm going to talk about the biomembrane structure, its constituents using fluid mosaic model. We know eukaryotes and prokaryotes are made up of cells, and all the machinery for metabolic activities is present in these cells. And if we talk about a single cell at a time, it simply means it must be excluded from the rest of the surrounding, or we can say other body cells. So it can be said this differentiation is necessary for what? To ensure the cell functions independently, and there is no mix up of the cell components with what? With the materials which is present around the cell or the outside of the cell. Now this exclusion or the separation or the differentiation of these cells is achieved through how? Through this biomembrane or cell membrane, or we say plasma membrane. And this plasma membrane serves some important functions. We discuss that functions first. So the first function here is it defines the cell and separates the inside from outside. What is this defining the cell means? In simple manner we can say it's informing the cell about its territory, that you have given this much space to work and carry out your functions, and you cannot simply mingle or mix up with the surrounding without need and without approval. And in doing so, it can be said plasma membrane separates the inside environment of cell from outside. And the next function here is it makes continuous barrier around cell. Must say it's a permeability barrier. From our knowledge, what barrier means? It's a stop. That means you can't cross me. Okay. Permeability barrier, or more precisely selective permeability barrier, means it allows the incoming or outgoing of certain molecules or ions and blocks the same, that is the exit or entry we can say, for particular substances. About these mechanisms and molecules we would discuss in detail when we talk about the transport mechanisms. So we can say this permeability barrier helps to maintain what? The characteristic difference between what? Between inside and outside of the cell. What does this mean? It simply means, in simple terms we can say, there is high concentration of certain molecule inside cell, and outside the cell it's very low. So this is making a characteristic difference between the concentration of that substance in these two portions. Let's go to the next slide now. This is about fluid mosaic model. Before we understand, let's split the term fluid, mosaic and model. So what this model is about? This describes the structure of plasma membrane as a mosaic of components. So this is mosaic of components. So these components means the structural molecules which are making this layer, this plasma membrane. So these are various components such as carbohydrates, proteins, glycoproteins, cholesterol, etc. You can see this in the diagram here. Here you can see this is glycoprotein, globular protein, integral protein, surface protein. In case of this glycoprotein, the protein is attached to a sugar moiety. These are responsible for signaling. Okay, and then here these surface proteins present on the surface of this plasma membrane. Integral protein, these are embedded in the biomembrane. And in case of this here transport protein, the protein channels which are responsible for the passage or the transport of certain molecules or ions. Okay, so these are some kind of different types of substances or the components you are going to see in the lipid bilayer. Now I'm going to tell you about the meaning of the second component here, mosaic. So this is simply the bunch of these structural components together, or their collaborative arrangement, or we can say the pattern and the pictorial representation of these biomolecules or simply molecules is referred to as mosaic. Okay, and the next point here is about the membrane, which is the phospholipid bilayer. This is bilayer, so it is composed of two layers. Let's talk about it in the figure. What do we have here? See this red color, this and the below one. These are constituting the lipid bilayer. They are composed of lipids, more precisely phospholipids, the lipids which are attached to phosphate groups. They have special properties, that is, they constitute two components. First is hydrophilic heads and the second is hydrophilic tails. About hydrophilic heads is that they are present in the extracellular matrix or the outside of the cell and the inside of the cell in case of cytoplasm. Why they are present there? Because they are going to love water. They are hydrophilic components. And the interior of this bilayer here, what you are seeing here is hydrophobic tails. So they are present here because they do not want to come in contact with water. So this is about the phospholipid bilayer. Next here is the question, why it's fluid? Why we call it fluid? What makes it fluid? And this is the most important, you can say, idea in phospholipid bilayer about fluidity. What we can say about this? We know there are various molecules which are present in bilayer. Okay, these proteins are actually floating around within this double layer, or in other words, it can be said some parts of the membrane, some parts, not all, that is, these proteins and other components can move around freely if they are not attached to other parts of the cell. So one thing you have to note here, not all proteins are free because many are attached to or embedded in the bilayer. Integral membrane proteins, for example, okay, they are embedded in the bilayer. What about then individual molecules of phospholipids? These molecules have the ability to diffuse rapidly within their own monolayer. So these phospholipid molecules, individual phospholipid molecules, can diffuse from one place to another in their own single layer, that is monolayer. And about this particular property of diffusion of these molecules, there would be a detailed experimental explanation in a separate video. So for now, we just know that bilayer is mobile or show fluid-like matrix. So from this explanation, what can be said? It can be calculated, it can be concluded or inferred that these moving molecules gives a fluid matrix. Okay, these moving molecules gives a fluid matrix that has the consistency of vegetable oil. Okay, so they have the consistency of vegetable oil, more importantly at body temperature. And now the next point is the proteins that are acting as channels. Okay, so these are some proteins through which there can be the passage of certain molecules like amino acids or ions, that is the transport of materials is carried out through them. includes active transport and passive transport. There is one more thing about cholesterol molecules. They increases the membrane fluidity, and we can say they are responsible for flexibility of membrane. So this was about the fluid mosaic model. In the next videos we would talk about the detailed structures and their functions of the each and every component